have an old an old friend, Diego Fontaneto, that works at the Italian CNR-IRSA, that is basically the same but without the S, so it's CNR, in the Water Research Institute in Piedmont, and he mainly works on speciation and evolutionary ecology on rotifers. And yes, I'm kind of I actually know Diego because he kind of guided me through my academical career. So yeah, this was just to thank him, and I will let him speak.
Okay, so I start sharing my screen. Here, I'm still in Italy and not there with you, but I will still talk about what we plan to talk about, and it's evolution and speciation when sex is not there. And I will mention one specific group of organisms that were notorious not to have sex and still to have species. But we start from far away. And often we can also recognize them. Like in animals, we may have males that are bigger, or we can have males that are smaller. In vertebrates, we can have animals that are both males and females, and both of them have sex as males and female, like the earthworms, or like the slugs, in which the winner becomes the male and wins over the other, who becomes a female. In flowers and plants, things are even more creative, with different individuals, different. But regardless, is thought to be a stable strategy, and that's having several small gametes, like sperms in males. So sex is all the process that drives the possibility. Oh, I see there are problems. Sorry.
Yes, Diego, I seem to have, several of us seem to have interruptions.
Okay.
So perhaps you can turn off your video and see if it's better.
Yes, I will try again. So stop video.
Let's try this way. Thanks.
Okay. Well, anyway, what I said until now, it's super boring. It's just that males and females exist. And it was the way to arrive to the fact that meiosis is the process that we know is creating variability in all the offspring, because the production of eggs and spermatozoans or pollen in plants is the mechanism through which we have the formation of offspring that are all different among each other and different from the parents. And this process of meiosis and sex that creates variability permeates all multicellular life as we know it. As a consequence of meiosis and sex, we have reproductive isolation when sex cannot happen. And the outcome of this is the biological concept of species, so that we have interbreeding animal organisms and produce fertile offspring, and sex is the main driver to originate and maintain diversity. This is the mainstream idea, even if speciation is a highly controversial issue in evolutionary biology. People fight a lot about details, and there's new tools coming out and new approaches to look at the process and at the patterns that we see in the field to understand better what are the peculiarities of what happens. But what we still see is that reproductive isolation that creates the interaction of gene flow between populations is what we all think is the main driver of the differences between species. Even in this seminal book in speciation, they deal only with reproductive isolation and gene flow. And there is one sentence saying, we don't deal with parthenogenetic asexual animals, organisms, because it's impossible to talk about speciation in organisms that don't have sex. And they kind of dismiss all the analysis on this specific group of organisms. Also because in 2004, nothing was really known about asexual animals. But what was already known is that this peculiar group of organisms, the bdelloid rotifers, they are known to be asexual. They are all females and they reproduce only through mitosis. So they produce eggs that are identical to the mother. And taxonomists recognized 450 species. So they were named an evolutionary scandal because they are asexual, but they have species. So in principle, they are a paradox because we know that without sex, you cannot have the division between species because there's no meiosis, no interruption of gene flow, and nothing of what we saw until now. So how to solve the paradox? There are three alternative hypotheses that we can start dealing with. The first one, obviously, is that it's not true that they are asexual. Maybe they have some hidden male that we don't know anything about. Or species do not exist. Names are there, but maybe it's just our tyranny of our state of mind that always wants discrete objects around us. And then we describe species of bdelloids, even if they are not biological reality. And that's the first half of my talk will go through these two hypotheses. If none of them is supported, then we can also make the inference that sex is not the only mechanism to have species. But let's start with the checking how do we know that bdelloids don't have sex. The main evidence is absence of evidence. The first bdelloids were seen by Leeuwenhoek when he invented the microscope. And then he could look at these tiny animals in the gutters of the roofs. And since then, only females have been seen. No males has ever been seen. So the first evidence we have is absence of males, which is not totally true because in 1823, a Danish zoologist published a paper in which he said that once looking at bdelloids in a frozen lake, he found one small dot moving from one female to the other, assuming it was a male, but then lost it and couldn't observe it. So that's the only potential indication of a male, but nobody believed it. And we have also indirect evidence of a potential lack of meiosis and of bdelloids being really asexual. There's a review from 13 years ago saying that they have accumulation of deleterious mutation, as expected if you don't have meiosis getting rid of deleterious mutation at the population level. Transposable elements move through sex, and in bdelloids, they are weird. They are not the usual transposable elements. And there's a long list of accumulating evidence of things that should be different if you are asexual. And in bdelloids, they are there. But they are there a bit, not so strikingly clear. So for example, all the genes that are involved in meiosis, they still have them. They didn't lose any of those potentials for meiosis. And also the accumulation of deleterious mutation. Yes, it's higher than in the sister group that makes sex, but it's not totally unbelievably different. Transposable elements, they still exist. They are strange, but they are still there. And so the genomic evidence, the genetic evidence is not so clear. But then in 2013, we got the first genome and the evidence was there. Like the chromosome rearrangement was strange. It didn't fit with meiosis. There were lots of palindromic repeats. Different alleles were messed up on different chromosomes. So apparently, that was the first true evidence of lack of sex, with really strange organization of the genome that is structured as a tetraploid with degenerate parts. So not everything is in 4 copies. And then some parts of the genome are more duplicated than 4. Chromosomes are not an even number. Strange things happening. And in that time, we were convinced bdelloids didn't have sex. So we can assume that they don't have hidden males, so they are really asexual. The other alternative is then they should not have species. They exist, meaning that we have 450 names. But as some of us know, if you are a taxonomist, you want to describe a new species every time that you find something strange. And maybe in bdelloids, there is a gradient of morphologies as expected with asexual reproduction. And only the ones that are peculiar for some reason get a name, and all the intermediate ones, taxonomists are not able to identify those animals and simply don't give a name. So we are creating diversity with different species as an artifact of our description of biological diversity. We can test it. We can test whether there is only one single species with a swarm of clones slowly diverging among each other through time, or if we have evidence of independently evolving lineages behaving like if they were species. And we can then look at some processes that could drive the, like geographic isolation or divergent selection. Focusing on one genus, that is a common genus living in water. All over the world, they are present. It's a good group because it's very easy to find the same species all over the world. And they have also a clear and homogeneous structure of the teeth of the masticatory apparatus that you see at the bottom of the photos with letter E. So that we can make a mathematical reconstruction with geometric morphometrics of the shape. And then we can compare whether shapes are really creating clusters from morphometrics. We can then look at the DNA data. In the ancient time when it was still Sanger sequencing, we were looking at one mitochondria and one nuclear marker to see whether there were discrete clusters. And on DNA, the discrete clusters seem to exist in a phylogeny, and they matched this name of the species that taxonomists could give to those organisms. So apparently, taxonomists didn't do a bad job. They were identifying clusters. Those clusters matched DNA. The same clusters matched also the shape of the jaws of the masticatory apparatus that was structured with discrete clusters. And then we have the first answer. Taxonomists didn't invent species. They look like phenotypic and genetic clusters that behave like normal species in sexual organisms. But still, we need some better support with more quantitative approaches to identify whether the clusters that we could see were indeed independently evolving entities behaving with different processes within than between clusters. And that could be done by different approaches in a phylogeny, checking, for example, whether there is a threshold between coalescent models within species and Yule processes between species, mimicking speciation, or with different Poisson distributions for branch length within and between species. There are some of these approaches in DNA taxonomy that can be used to identify potential species. And in all the bdelloids that have been looked until now, those methods matched the idea that those clusters are indeed independently evolving entities. So species exist, clades exist, and they are really independent. The second step now is, do we fit with our idea of isolation? If one starts looking at different habitats and checking different species, the different species have a very species-specific habitat in which you can find them. So apparently, the field studies support the idea. If you also do genetic reconstruction of diversity in space, the different habitats have different clades. They even have competition between species. Looking at the belly, the bottom part, the lower part of these freshwater crustaceans, you can find up to 10 different species of rotifers, of bdelloids. And the distribution, the probability density function of the distribution of those species on each crustacean depend on the co-occurrence of other species of bdelloids on the same individual. So for example, there is one species that loves staying close to the mouth, but it can do that only if it's the only bdelloid living on the crustacean. Because if there are other competitors that are stronger, they push the losers far away from the mouth. So they do have ecological niches, even if they are asexual. And those niches are both abiotic and biotic. We can make it even more supported with a quantitative analysis by checking the neutral expectation of morphological variability within what we know from neutral markers from the genome. And then we can look whether the geometric morphometric representation of the jaws produces variability that is neutrally evolving, or it's more clustered, significantly more clustered than expected. And it's always the same idea, that jaws are more fixed in the shape than expected by chance, because they have to do their job to help the animal getting food. So there is selection acting on the shape of these jaws. Ecological selection exists. Biogeography is a bit more tricky, because wherever you go in the world, you find the same species. There's only Antarctica that is different from the rest of the world in bdelloid species. But the rest of the world, where we have the same species, if we look at DNA diversity, there is a signature of a correlation between geography and genetics. So geographic isolation can work also in bdelloids. Not so strong as in other organisms, because these animals like bdelloids are able to desiccate and fly with the wind, and they can remain ages desiccated, so they can be moved all over the world. But for us, what we arrive now at this step is that species exist. Anything you can try to find out to identify clusters that make sense according to what we know how species should behave, it's there. And then, if we know that they are asexual, and if we know that species make sense, the only consequence is that sex is not the only process that can create speciation and can originate species. This is what we knew in 2015. And that was this is the explanation that we had. Is that if we go back to the general theory of how species exist in sexual organisms, geographic isolation and divergent selection are the first drivers of the diversification, on top of which reproductive isolation occurs between populations, creating genetic and phenotypic clusters. If we remove reproductive isolation, the idea can still hold. And actually, if we remove reproductive isolation from the scenario, we arrive at a point in which we can even think that without sex, speciation is faster, because reproductive isolation is a hindrance to the production of phenotypic clusters and genetic clusters, because they mess things up all the time if gene flow exists. If gene flow is out of the game, and we have only genetic divergence created through divergent selection and geographic isolation, speciation can go faster without sex. This is not novel. 40 years ago, it was already demonstrated mathematically that it could be. And then we try to show it empirically. Is it true that the asexual rotifers speciate faster than the sexual rotifers? So we started looking at the first things like amount of cryptic species. How many genetic entities that are separated with statistical support are there within each of the nominal taxonomic species that we can identify from morphology? Interestingly, all the species that were analyzed with this idea in mind came out with cryptic species. So there's more diversity from the DNA than expected from morphology. Diversification. Unfortunately, we discovered that they have sex. All the species that we looked at, they have cryptic species. So the idea of higher genetic diversity than morphology, sex yes, sex no, but it's topic animals. Because if you are, there's no way to give a name of a different species again. So morphology is not a good proxy for diversity for really small animals. Sex, all the five that cryptic species. Higher level of but we can look at
Diego, sorry.
Diego, are you still with us?
Yes.
The connection is really choppy. Do you by any chance have another way of connecting to the internet?
I will try now with that. Sorry. So
sorry for that.
You're welcome. No problem. Okay. Now I start again.
Let's see. Okay,
Yeah,
thank you so much, Diego.
No, sorry. That
you can perhaps go a few slides back. Perhaps, let's say, can you go back a little? I'll let you know again. bit more. Yeah, just the last slide, the one on the
Okay.
cryptic speciation.
Yes.
I think was fine after this one. Sorry. Yes, when you got from the first to the second, this is where we really lost you the most.
Okay.
No, just repeat what you said.
Say that the relatives of the bdelloids are the monogonons. They have sex. They are rotifers. They live in the same place. The only difference is that bdelloids are potentially asexual, and the monogonons are not. They do have males, and they produce meiosis. There's a same level of cryptic species, which is a pattern in common with all these microscopic animals, for which morphology is not a good proxy of diversity. So we are still in trouble, because if we want to demonstrate that speciation is faster without sex, cryptic species is not a good route to go. But we can check more refined patterns in the diversification process that we can mimic through phylogenetic reconstructions. For example, lineage through time plots and a representation of how fast diversification occurred and whether it changes through time, being faster in the origin or faster towards recent times, with the idea that the expectation is asexual should go fast at the beginning and then slowing down according to ecological speciation models in which niches are filled, and then it's difficult to have new species. But at the beginning of a diversification event, it should be faster. And that's exactly what happened, that in all the groups that were analyzed of bdelloids without sex, the lineage through time plot is decreasing according to what was the expectation without sex. And the monogonous rotifers, the ones that have sex, diversification rates are constant through time without deviations. So apparently, in this case, sex is important to create rates of diversification. Then we looked in detail at other metrics, for example, how much diversity is there within each species, how much diversity is there between closely related species, how old is the most recent common ancestor of each species, and how old is the node splitting the two closely related species. And here again came out that sex and no sex, genetic diversity within species, the same. Time to the most recent common ancestor, the same. But in bdelloids, the node between two closely related species is always much more recent than in monogonons that have sex, in which speciation takes much longer, even within closely related species. So this was, again, the only suggestion that indeed speciation can go faster without sex. And sex was considered a hindrance for diversification. We were happy because we thought we could understand the system, but there's always something coming in from the window. And that's the fact that bdelloid rotifers not only are notorious to be asexual, but I already mentioned that they are able to desiccate and survive freezing. And when they are dry or frozen, they can survive a long time. Wind can displace them all over the world. But what happens during desiccation is that the DNA physically breaks. And when we take a desiccated animal and we put it back in water, in 5 to 10 minutes, the animal is happy and live again, feeding and moving around. And within 1 hour, DNA is not broken anymore. So they have a super fast and efficient DNA repair mechanism that allow these animals to repair DNA. This was already known, but what we didn't know is that through this mechanism, potentially, they may be able to incorporate genes that are not from the animal. And now we know that 10% of the genome is not animal. So within their genome, they have genes that come from bacteria, fungi, plants, protists. Not only we know that they are not animal, but we can also track bacterial genes that became animalized with a different ATGC content. And there are bacterial genes into the genome of bdelloids with introns that could then take totally different functions from the original genes when it was still bacteria. So the level of horizontal gene transfer that we know on all the species that have been looked until now is always within 8–10% of what is not animal. We don't know if there are genes that could come in from other animals because we cannot really be sure that they are not originally already from the bdelloids or horizontally transferred. But with these other non-animal genes, we are sure and confident that they are there. And it's not due to contamination, because you can really track that those genes have bacterial origins, but they are not bacterial anymore because they function as if they were in animals. Something interesting that came out here and that made us support the idea that horizontal gene transfer happens during desiccation is because when we compare transcriptomic data from different bdelloids, comparing bdelloids that are able to desiccate with bdelloids that cannot desiccate but can live only in permanent water. If we look at the graph in the middle, proportion surviving desiccation, the two species, sword and tard, the two at the bottom, that can really survive a lot desiccation, have also a much higher proportion of transcript that are horizontally transferred. The two species that do not desiccate, Mag and Sochi, they have a very much lower proportion of transcripts that are horizontally transferred. Thanks to this analysis, we also tracked how often one horizontal transfer gene can enter into the genome. And with some huge approximations, it could be within every 50,000 to 100,000 years. So it's true that horizontal transfer is common. 10% of the genome is not animal, but it's not so super common. So it's once every 50,000 years, which should not be enough to create variability and to allow speciation. It's really not enough. But then we started looking at additional things that were, can horizontal transfer happen not only with bacteria and protists and plants, but can it happen between species of rotifers? And then again, we checked different mitochondrial and nuclear markers in a small population of a species in a park in Belgium. And yes, we found evidence of things that didn't match. Mitochondria of one group and nuclear markers of another group. We went even further, asking, can this transfer happen within species? And yes, looking at different markers, we found triplets. So like groups of 3 individuals in which the arrangement of these markers could be explained not by common ancestry, but only by exchange between individuals. And this is true for all the markers that we looked at. Then what we suggested is that, well, of course, sex may not be the only driver of speciation. If we have alternative ways of making sex, or exchanging genes like horizontal gene transfer when the animals go desiccated. We were really happy because the picture we had in mind started making sense. All the additional analyses that were coming in fit with the idea. But then no, like few other analyses show that if you really look at the genome better, comparing different species and making better genomic bioinformatics, then there's no evidence of asexuality in the genome, like the palindromic repeats disappear. The composition of in the chromosomes, they were artifacts and not evidence of asexuality. And if you compare desiccating and non-desiccating species, the level of difference in the horizontally transferred genes is the same. So the transcriptomic differences we saw were maybe related to the habitat where the animal lives, that the ones that live always in water live in a different habitat than the ones that desiccate. So the transcriptomic differences were ecological and not to the amount of horizontally transferred genes. And also the population genetics analysis with horizontal transfer genes within and between species, they were most likely explained by contamination. So in this case, the evidence was a mistake. And we are back to our hypothesis of, are they asexual? We don't know. So we know that species exist. That's still okay. But everything that was built up to understand whether they had sex, they can have sex or not, now it's in doubt. And since 2018, the paper I showed you before, more papers came in. If you want to know more, there is a book that was published in 2019 with the title Evolutionary Biology of Species. The idea is to talk about all species, but there are bdelloid rotifers on the cover. So of course there is a lot mentioned on bdelloids. But even if you read this book that is relatively recent, the story is not at the end, because then the following year, this paper came out saying there is genomic signature of recombination in a population of bdelloid in which they analyzed 100 individuals and they checked comparative genomics. And they show that there is evidence of recombination in these animals. Same species, Adineta vaga, another group looked at one single lab clone, but reconstructing chromosome level genomes better than before, and they found that there is no evidence of asexuality, at least not strong. Another species and an approach that goes in between population genomics and better chromosome reconstructions, again, they found signature of sexual reproduction, potentially. And now we are again at the level of which we may have something strange. So sex may exist, or at least genetic exchange may exist, but maybe not through meiosis. And the current work that is carried on bdelloids at the moment deals with two aspects. One is trying to understand the mechanism through which they can exchange genetic material at the species level so that we can understand whether meiosis is involved or not. And the other line of research is doing more population level studies to really understand the effect of this genetic exchange and the magnitude of it. So if it's only small parts of the genome that can have this recombination, or if it's all the genome. And now I conclude, going back to my first slide with the title. The only difference is that there is a question mark at the end, because the without sex, we are not really sure that is true. So we still don't know whether they have sex or not. If they have genetic recombination, is this similar to meiosis and sex as we know it? Or do they have some science fiction ways of doing genetic exchanges? A few years ago, a guy from US, Matthew Matheson, suggested the term lesbian necrophilia with the idea that two bdelloids can exchange genetic material when they go desiccated. One of the two dies. When dying, releases DNA in the environment. Water comes again. The one that is alive resurrects. By resurrecting, fixes its own chromosomes using the genetic material of the friend that was there and who died. And then they exchange genes in this peculiar horizontally transferred system with the requirements that both of them are females and one of them have to be dead. And that's why the term lesbian necrophilia. Now we don't know if this is true or not. And more line of research are going on to understand whether this can be supported or not. Okay. So now I stop sharing and I will start my video again, hoping that the internet connection will be stable enough.
Thank you, Diego. Yeah, it was fine for all the rest of the presentation, so no problem. So we still don't have questions in the Q&R, but I can ask a few ones in the meantime, or I'll come up. Here's one. I hope you don't get into many spams with your if you have emails that are talking about sex and lesbian necrophilia, but here are some questions that are raised.
Well, when in 2007 I published my first paper on species without sex in bdelloids, it got a good press release. Yes. For science, but not for me, because the typical sentence was, how to survive without sex, asked Diego Fontaneto. Or millions of years without sex, here the researcher who discovered it. And okay, it was fun for me, but being Italian in the UK, the jokes were really...
I can imagine. So Mike had a question. Mike, it's a simple question, but you can ask it. Perhaps you want to interact with Diego a bit more.
Can you hear me?
Yes.
Okay. I wanted to know if it was possible to culture this species in a lab setting. So could you test to see if horizontal gene transfer is happening? Could you do a desiccation experiment where you added an external rotifer DNA, and maybe see if you could figure out the recombination hypothesis in that way?
Yes, that was when horizontal gene transfer came out. That was one of the first things that people tried. So like you do a PCR amplification of a gene that you know, and you use the PCR product as the liquid in which you revive a bdelloid rotifer that was desiccated, putting a huge amount of a marker that you know was not there in the bdelloid, and then checking whether you will find it later. And no, it never happened. So all the time that people tried these or other more refined ways to try to expose a dry rotifer to foreign DNA and to force the horizontal transfer, it never happened.
Okay.
The reason could be that if it's really true that it's a successful horizontal transfer every 50,000 years, the likelihood that you can find it in your lab, it's really negligible, that you need to be super lucky.
So you could think that with the PCR and the amplification, you would have increased this likelihood, no? Yeah,
that's what people were doing. Like you put a huge amount of DNA, increasing the likelihood. And then they also tried with the plasmids, like the way that bacteria would do it, but still it didn't work.
Okay, thanks.
You're welcome. There is Paul.
Yeah, hello. Thanks for the talk. So you mentioned the possibility of speciation in a context of mitosis, but the recent papers, they tend to show that recombination is there. So probably meiosis, maybe modified meiosis. And how would that change the idea that diversity could be produced in that way, still with no fertilization if males do not exist? And how could that allow or not for speciation?
Well, it's a very good question to which I have no answer. And I think the answer is only creativity from the side of the scientist that can try to come out with a potentially science fiction hypothesis that then you want to test on your organism. Like screening what happens in the other organisms that we know have weird meiosis, like the Enotera style, in which chromosomes don't pair, but they attach only at the telomeric regions, so that you can allow for a combination even if it's not complete. There were different tests that were done, checking what is known in other organisms, and then checking whether it was there in the bdelloids. But until now, everything maybe sometimes pops out as a new idea that seems to fit. Like we had also the Meselson effect that seemed to fit for a few years and then disappeared. The lesbian necrophilia idea came out and then it's out of the game because people don't believe that it's really horizontal transfer, the driver. So we are in this limbo in which different labs are trying to test different hypotheses. And until now, no one yet discovered how the genetic recombination happens. So there's an ample field for new ideas to come in from young people and to test if you can really find an answer.
Thank you.
Well, sorry, so I had no answer. Yes, Mathieu?
Hi, hi Diego. I was just wondering what we knew about mutation rates in bdelloid rotifers and if that could, how does that compare to other microorganisms which reproduce asexually?
Yes, everything is only relative. So it was tested with some mitochondrial markers and then heat shock proteins and some alpha tubulin markers, comparing different species of bdelloids and different species of monogonods. But then nothing is really so different, apparently. We don't have absolute mutation rates because there's no way to date a phylogeny of bdelloids, of rotifers in general. A way should come out soon. Like we have colleagues who are working on parasites of bdelloids. And those parasites are fungi. And those fungi are known also from, there's a huge record of fossils of those parasites from arthropods, from amber, and from other organisms. So those fungi, we know when they phylogenetically invaded bdelloids. And they invaded bdelloids 60 million years ago. If this is true, then bdelloids may be 60 million years old, roughly. Other evidence using clock models based on evolutionary rates from other invertebrates, they fit into the 40 to 100 million years. So rotifers could have that age, and bdelloids also a bit younger than 100, maybe just 80 or 40. But even if you use these boundaries to try to estimate evolutionary rates, then the numbers that you get have this margin of errors that are covering almost everything. So we cannot really pinpoint the true evolutionary rates at the moment. And people who are trying to do that with lab cultures and trying to really see in action evolutionary rates, they were not successful until now. So it's something that I'm quite confident will come out soon with new genomic analysis at the massive screenings, but we have no answer for that at the moment. Sorry.
And there's no rotifers in sediments or things which could be younger
You mean rotifers
and sequenceable that you could resurrect from sediments? Or...
Oh, yeah. The last year they found a rotifer from 24,000 years ago in Siberia in ice. It was a frozen block of ice in permafrost, and the bdelloid rotifer came out. So that bdelloid is 24,000 years old. And they are sequencing it now. They have it in culture, so it's growing. Russian people are working on it. But again, can we really trust that the animal was 24,000 years old, or it's a recent contamination? There's still a huge debate on that. I hope it's true that it's very old, but it's not so clear. And the oldest from sediment that was resurrected, I think it's 100 years old. The oldest known and proven. And maybe in 100 years, they didn't really accumulate much mutation. And so no way to answer it at the moment.
Okay, thank you.
Okay, we're going to move on to Patrice David. Also, taxonomical question, I guess.
Hi, can someone hear me?
Yes.
Yeah, hi. Yeah, I have a question about, it's not really a taxonomical question, it's about all these problems of defining a species in asexuals. And I think most of the time, and I think that's what you did at the beginning of your talk, the question of whether species exist in asexuals is slightly changed into, are there any consistent and non-arbitrary clusters that are supported by both morphology and genetics in asexual groups? And I think the answer is clearly yes, there are clusters, but I think the question is slightly different to whether there are species. I understand that these clusters have been given species names by the taxonomists since ages, and they were right to give them different names because they are actually different. But the question is, do we mean the same thing when we say these are species, and when we say this or that sexual group is made of species? And because in sexual groups, we have species which are genetic and morphological clusters, which are supposed to be held together by sexual reproduction. I mean, they have their consistency because of sexual exchanges. But we also have higher rank in taxonomies, which are not arbitrary at all, like families, genera. Usually taxonomists do believe that they have some value. They are not arbitrary groups. So why do we equate the morphological and genetical clusters we see in asexuals? I say asexuals because I think it's not only rotifers. We hear that about bacteria or other predominantly asexual groups. Why do we equate these clusters to species, not to a higher taxonomical rank?
No, you're totally right. I think it's just a matter of how would you define species and what your philosophical approach into that. Because you are absolutely right that in the past, people thought that only species were biological realities, and then genera, families, and higher ranks were just convenient groups until you reach phylum that maybe was another biological reality. But then if you test with these methods that we saw in DNA taxonomy with the coalescent and Yule or Poisson processes, and you test higher ranks, they often come out as actual biological entities. Also families, within family, the processes are different than between families. So they make sense. And you're right that if I use the same method, the only difference is that to reach the processes at the level of families, I can take only one sequence for each genus. Well, one individual for each genus in my reconstructions.
Mm-hmm.
If I want to check genera, I take only one individual for each species.
Mm-hmm.
When I want to check if they are species, I use all the individuals I have. And then I can assume that if I have a grouping of individuals that make sense genetically, morphologically, and ecologically, it should be the lowest rank we know in diversity, that is species.
So this, then you assume that the right definition of species excludes reference to sex and is the lower grouping that we can make and which is consistent with morphology and yeah, because if I mean, for me the alternative was not that there would not be species in asexuals. The alternative was that every individual would be a different species in asexuals. If you take the usual like old Mayer definition of species based on reproductive compatibility. Yes. So, and it's probably a matter of convention, but yeah, I think what the bottom of all this is that we do have some kind of unevenness in the way lineages diversify, and the only cause of not being an even process, being something quite irregular with different levels of hierarchy, is not only sex. That I think we can agree all about.
Yes, no, but you're right that the term species can be misleading if you equate species as the product of meiosis, and then if you don't have meiosis, you cannot use species.
Yeah.
You should invent a new term, but then it can get even more complicated and more questionable, the term that you would use. So I think people call them species just for convention and to make them understandable with the others. That's the level you are dealing with, and without really making too much philosophical.
Yeah. So yeah, in philosophical terms, it would be a kind of disjunctive definition that you take different definitions depending on the examples.
Yes.
But yeah, in practical terms, I understand that everybody uses species anyway. So.
But I think this would be a large debate. I remember some papers by Jim Mallet a long time ago that were saying that that's what Darwin actually meant. He was aware of hybrid incompatibilities, etc. He just thought this was not the most important aspect of the definition of a species. So I think these people still debate these issues to some extent, if I may. Just to react before we take the last question, also I was puzzled that you consider that somehow sex is required to maintain the clusters in a way, because it seems, from what I understand from models, and you wrote about some ecological explanation based on competition, for instance, from what I've seen in the theory, when you have disruptive selection that's caused by competition, for instance, you readily get clusters that are stable, etc. In asexuals, what's more complicated is to separate them in sexuals, but in asexuals, if you have an ecological explanation of disruptive selection, then you expect to have some clusters in a way, right?
Yes. Yeah. Actually, the difference is that in asexual, you expect some cases of clustering, but also a lot of swarming without really clades, because you cannot always have competition and always have disruptive selection. You may have some neutral things moving around anyways.
Especially for cryptic things that have more or less the same thing.
Yes.
So we have a question by Christoph Haag.
Yes. Hi. Can you hear me?
Yes.
Okay. Yes, I wanted to ask. I'm not totally sure if I may just have missed it, because I think at that moment my connection wasn't very good. But you seem to have this observation that there was a slowing down of the speciation rates in bdelloids, whereas it remained constant in the monogonons. And I'm not totally sure I understood the data pretty well that on which this observation was based. So that's my first part of the question. Second part of the question would be, so then you had this other observation that nonetheless the bdelloids had more shallow nodes between species. And so I was wondering how you could, do you have an idea how these two observations can be reconciled?
Yes, no, you're totally right. The two observations are totally in the opposite direction, because you would expect, if there is a slowing down of a diversity, you would not expect shorter distances. But what, well, first, what the data was a species within a genus. So all the examples are, we take a genus, we sequence all the species we can, as much as we can, to have a good coverage of the genus, and then we check diversification within the genus in bdelloids and in monogonons. And the patterns were consistent. I didn't expect that to be so consistent. And the slowing down is something that is predicted by ecological speciation when only ecology is there and not sexual reproduction, well, interruption of gene flow. What seems to be is that when we check the hypothesis of a closely related species, how close they are, we always took the most closely related species we had in our phylogenies. And it's likely that in the short, really short term of a closely related species, bdelloids are faster, and we have evidence of that. But all the species within a genus, it's still possible that there is a decrease in the speciation rate. It's simply that out of 50 species, we took 2 pairs of very closely related ones. Because if they are not very closely related, they wouldn't pass our filtering to check how closely related they were to analyze them. And by checking this in similar data set in bdelloids and monogonons, in the monogonons, the most closely related ones were still more distantly related than in the bdelloids.
Okay, thank you.
You're welcome. Thanks.
Okay, great. Well, with this, I'll just thank Diego again for a super stimulating talk and gave rise to interesting discussions. By the way, if people want to chat a bit more with Diego, he would be available this afternoon. You just need to contact him or Riccardo Poloni. His name is on the, I guess he's on the announcements. And so next week, we'll have a seminar at 11:30 again in Visio. It will be Anna Lockmer who will talk about protozoa in our guts. So with this, have a nice weekend, everyone. See you next week.
Bye-bye.